Hello everyone, this is me Ahmad Fathy from 12B4.
And Akram Fathy.
And I am Abdelraouf Moataz.
Today we are gonna present our DNA replication, okay, and also we have with us.
And I am Adam Mostafa.
DNA replication is a biological process by which a double-stranded DNA molecule is copied to produce two identical DNA molecules. It occurs during the S phase synthesis phase of the cell cycle, prior to cell division. DNA replication is essential for cell growth, development, and the transmission of genetic information from one generation to the next. Overall, DNA replication ensures the faithful transmission of genetic information from parent cells to daughter cells. It is a highly accurate process with an error rate of about one mistake per million nucleotides replicated.
DNA replication is of paramount importance for various biological processes and the maintenance of genetic integrity. Here are some key reasons why DNA replication is essential. First, genetic reproduction. DNA replication is crucial for the reproduction of genetic material during cell division. DNA repair and maintenance. DNA replication also plays a critical role in DNA repair. It provides an opportunity to identify and correct errors or damage in the DNA sequence. Third, protein synthesis. DNA replication is directly linked to the reproduction of proteins, which are essential for the structure and function of cells. Fourth, genetic variation and evolution. Introduction of genetic variation. Although replication is generally accurate, oscillation mistakes can known as mutation can occur. Fifth, diseases prevention and diagnosis. Understanding DNA replication is crucial for diagnosing and understanding genetic disorders. In summary, DNA replication is vital or for genetic reproduction, protein synthesis, DNA repair, genetic variation, and disease prevention. It ensures that accurate transmission of genetic information and plays a fundamental role in the functioning and development of living organisms.
Number one, proofreading by DNA polymerases. DNA polymerases, the enzymes responsible for synthesizing new DNA strands, have built-in proofreading capabilities. Number two, mismatch repair. Cells have additional systems known as mismatch repair pathways that help identify and correct errors that escape the proofreading capabilities of DNA polymerases. Number three, DNA damage repair. DNA replication errors can also occur due to the damage from various sources such as radiation or chemical exposure. Number four, cell cycle checkpoints. Cell cycle checkpoints act as a control points during the cell division process, including DNA replication. Number five, DNA damage response. Cells activate various signaling pathways, collectively known as DNA damage response, when they encounter replication errors or DNA damage. It is important to note that while these mechanisms reduce the occurrence of replication errors, they are not perfect. Mutations and errors can still arise, leading to genetic variations and potential health implications. However, the combination of proofreading, repair system, and cell cycle checkpoints significantly minimizes replication errors and ensures that faithful transmission of genetic information from one generation of the cell to the next.
First one is initiation. DNA replication begins at specific sites called origins of replication. Proteins called initiator proteins bind to these sites and spread the DNA strands, forming a replication bubble.
Unwinding. Enzymes called helicases unwind and separate the double-stranded DNA by breaking the hydrogen bonds between the base pairs. This action creates two replication forks, where the DNA strands are exposed and available for replication.
Primer synthesis. Before DNA synthesis can start, a short DNA primer is synthesized by an enzyme called primase. The primer provides a perfect starting point for DNA polymers to begin adding nucleotides.
Number 4. Elongation of leading strands. DNA synthesis occur in a 5 to 3 direction, meaning new nucleotides are added to the 3 end of the growing DNA strand. DNA polymerase 3 binds to the primer and starts adding complementary nucleotides to the template strand in a continuous manner. This process occurs smoothly along the leading strand toward the replication fork.
Fifth one is elongation of lagging strand. The lagging strand is synthesized discontinuously in short fragments called Okazaki fragments. As the replication fork progresses, DNA polymers 3 adds nucleotides to the lagging strand but in the opposite direction of the replication fork. When the polymers reaches the end of an Okazaki fragment, it detaches and moves backward to synthesize the next fragment.
Okazaki fragment processing. After the synthesis of Okazaki fragments, the RNA primers are removed by an enzyme called DNA polymerase I, which replaces them with DNA nucleotides. DNA ligand then seals the gaps between the fragments by forming phosphodiester bonds, creating a continuous strand.
Number 8. Proofreading and repair. Throughout the replication process, DNA polymerases processes proofreading capabilities. They can detect and correct errors in DNA synthesis, increasing the accuracy of replication. Additionally, there are DNA repair mechanisms that fix any mistakes or damage that may occur during replication.
Termination. The replication process continues until the entire DNA molecule is copied. When the replication forks meet, additional enzymes ensure that the DNA strands are properly sealed, and any remaining RNA primers are removed and
Good
replaced
morning, miss.
with
This is our
DNA.
project about DNA replication, and we are using in this project soap, salt, hot water, cold water, banana, isopropyl alcohol, and hand blender. And this will be done by Mostafa, Abdelraouf Moataz, Ahmad, Fathy, and Akram Fathy. First, we're going to add the salt and some soap.
And now we'll put some hot water. And now we'll mix them. And now we're going to add the banana. And after that some water. And 5 teaspoons from the liquid we did. After that we're going to blend them. That's it.
And now we're gonna filter this liquid here. And now we're gonna put the result of what we filter in the cup. And we're gonna add the isopropyl alcohol.